好，2015年第7條題目係講實驗嘅。今次講實驗之前咧，有2樣嘅化學物質咧要講下先。第一樣咧就係過氧化氫，或者平時我哋講嘅雙氧水啦。咁其實咧佢就喺我哋身體嘅一啲新陳代謝嘅反應咧，就產生出嚟嘅一啲副產物嚟嘅。咁佢本身有毒嘅。當佢漸漸積聚嘅時候咧，其實係會對我哋嘅細胞咧有傷害，長遠咧亦都真係會殺死一個人嘅。咁但係好彩地咧，我哋嘅身體咧就有一種酶叫做過氧化氫酶啦，就係能夠加速翻拆解雙氧水就變成水同埋氧氣嘅。咁呢個就係嗰條 equation 咧，俾大家睇下先。咁其實啦，呢個咁樣嘅化學物質過氧化氫啦，同埋過氧化氫酶啦，喺我哋嘅課本咧都係有講到嘅。除咗動物嘅組織咧，植物嘅組織都有嘅。所以啦，當初個實驗咧就講過話蘋果啊、薯仔啊、動物嘅肝臟啊、肉類啊，其實都係有嘅。咁今次嘅科學探究咧，就係想去調查一下溫度對於過氧化氫酶呢隻酶嘅活性究竟有啲咩嘅影響啦。咁下面咧有5步嘅，就係個學生咧要做呢個實驗嘅一個輕輕嘅草稿啦，啲步驟應該點樣做啦。咁啊1、2、3、4、5咧詳細嘅概念咧，我哋會喺 Part B 度講多啲嘅。而家咧我哋先講第一步。第一步咧就係喺一啲動物嘅器官咧去抽取過氧化氫酶，咁就關 Part A 事啦。Part A 就講啦，咁究竟我哋由啲乜嘢嘅動物器官去抽取呢啲個過氧化氫酶咧？而當中有個提示俾你嘅，就係呢一個嘅動物器官咧，佢係有好多嘅過氧化氫酶，所以我哋先能夠咁樣抽咗出嚟啊嘛，容易啲啊嘛，係咪？咁你話啦，有其他組織咧好少嘅，咁你抽唔抽取到？抽取到難啲咯，少啲咯，係咪？跟住啦，就去解釋下咧，點解呢個器官有咁多嘅過氧化氫酶啦？咁其實呢條題目咧就係考下我哋對於過氧化氫酶啊，喺我哋呢啲動物組織啊、植物組織啊、邊款組織係有多啲咧？咁其實書本正正呢個實驗咧，就係話俾我哋聽，肝臟係零舍多。所以今次考嘅基本概念咧，就係肝臟嘅功能啦。肝臟喺我哋身體的確好多功能嘅。喺消化嗰課咧，就係製造膽汁啦；喺體內平衡嗰課咧，就係講調控翻個血糖水平啦，儲存個糖原啦。關今次嘅題目事咧，就係解毒啦。咁咩叫解毒咧？對於一啲中等毒素嘅物質，例如酒精、例如藥物啦。前嗰年嘅題目咧，我哋都有講到噶。酒精同埋個紅菜頭啊嘛，原來酒精係可以破壞細胞膜。咁其實我哋飲酒精，當酒精濃度太高嘅時候，會唔會都對我哋嘅細胞有傷害呢？當然會啦。另一款啦就係藥物啦，藥物啦，你話攞嚟殺細菌咁梗係好啦。咁但係啦，殺得細菌嘅，對於我哋嘅細胞，莫非真係完全冇影響咩？都冇可能嘅。咁呢兩款咧就係最典型嘅有毒嘅物質，只係爭在佢一食唔係即刻毒死你。所以啦，有多有毒嘅物質咧，唔係毒唔毒得死你嘅問題，唔係 yes no 嘅問題，係 more or less 嘅問題。食少少定食多多，食少少都整得死你嘅嗰啲咪劇毒咯，係咪？咁今次啦，過氧化氫或者雙氧水都係一種有毒嘅物質，但係我哋身體咧就能夠解毒咗佢啦。所以答案咧就係肝臟。咁啊，肝臟嘅其中一個功能咧就係負責去解毒啦。又或者你話肝臟咧係一個好多新陳代謝發生嘅地方，咁其實佢都係會產生過氧化氫嘅。不過唔緊要，佢自己識得拆解翻，咁就冇問題啦。去到 Part B 咯，睇翻1、2、3、4、5 5步嘅實驗咧，佢就講啦，呢個學生漏咗一個好重要嘅步驟。究竟呢步係咩呢？呢一個步驟又有啲咩嘅重要性呢？咁我哋先睇下佢嘅呢5步究竟做咩先。第一步啱啱講咗啦，由動物器官抽取過氧化氫酶，然後就將呢啲過氧化氫酶就倒落去個雙氧水個樹，然後啦就擺佢喺0度啦，跟住就量度下喺頭5分鐘有幾多嘅氧氣係產生咗啦，然後啦重複2至4步，就喺20度、40度、60度。咁既然啦呢一條係科學探究嘅題目嚟嘅，咁梁 Sir 一定同大家溫一溫書先啦。整個實驗嘅 IV， 我哋想去調查嘅就係溫度，係整個反應喺乜嘢溫度發生呢？0度、20度、40度、60度，我哋轉變噶嘛，係咪？咁呢個 IV。DV 咧就係我哋想去觀察嘅，我哋就想觀察翻溫度對於過氧化氫酶嘅活性究竟有啲咩嘅影響。咁但係啦，我哋點樣去觀察過氧化氫酶嘅活性呢？就係透過觀察頭5分鐘能夠產生幾多嘅氧氣啦。呢個就係 DV。咁去到控制嘅變項呢，就例如啦，雙氧水嘅多少啦。咁啊，每一個 set up 咧，0度、20度、40度、60度咧，都應該要一樣嘅。咁除咗佢之外咧，仲有其他嘅，不過今次講住一個先啫。咁整個實驗啦，差啲咩嘢呢？咁睇上嚟1、2、3、4、5。都好正路啊，係咁樣噶啦，倒完落去咁咪擺一個特定溫度咯，係咪？其實啦，有一個好關鍵嘅步驟咧，我哋要留意嘅，就係我哋要 make sure 我哋嘅過氧化氫酶同埋雙氧水 當佢哋去溝埋之前，就已經要達到一個特定嘅溫度，即係話啦，喺溝埋之前，佢哋已經要係0度溝埋，咁咪0度咯。幻想下而家雙氧水係0度嘅，但係如果過氧化氫酶唔係0度嘅話，例如佢喺室溫25度嘅嘢倒落去一個0度嘅嘢，咁最後尾嘅混合物係咪0度啊？冇可能係0度嘅。佢係高過0度，咁即係話啦，你唔係度緊0度發生嘅事啦。咁你話唔係啊？咁我擺多一陣，佢都會變0度噶啦。但係當佢真正變0度之前，佢唔係0度啊嘛。但係個反應已經發生緊噶啦嘛。咁即係話啦，你個實驗係根本唔準確嘅。咁所以啦，你要去做嘅呢，就係將過氧化氫酶同埋雙氧水喺溝埋之前，擺喺一個特定嘅溫度。擺幾耐？例如10分鐘啦。咁就可以確保啦，兩款嘅物質都已經去咗0度，再撈翻埋一齊，咁就一定係0度啦，一定係20度、40度、60度啦。咁所以答案咧就係講緊將雙氧水同埋過氧化氫酶喺一個特定嘅溫度擺10分鐘，先可以去將佢溝埋一齊。咁呢步有咩嘅重要性啊？係確保喺整個混合物喺佢成個反應開始之前，就已經去到一個特定嘅溫度，例如0度、20度、40度、60度。跟住啦，去到拍試。今次嘅題目咧就考我哋畫個 set up 出嚟啦。呢條題目咧同過往唔同嘅，過往咧就俾個 set up 你啦，跟住問下你啦，呢個 set up 有咩錯啦？呢個 set up 有咩嘅重要性？點解要做呢一步啦？好似頭先嗰條咁樣，佢漏咗步喎，嗰步咩嚟嘅咧？又或者下次你又問翻你啦，喂，佢做咗呢步啊？呢步好重要㗎。咁究竟呢步有幾咁重要咧？呢條題目咧係好考我哋 SBA 嘅技巧嘅題目啦，俾咗一堆嘅儀器啦，同埋一啲材料我哋啦。咁究竟我哋點樣砌到個裝置出嚟咧？呢條題目啦，考兩樣嘢。第一樣就係科學探究，就係點樣去整個裝置。第二個就係科學嘅繪圖啦。咁啊提定大家先啦，畫得個裝置咧一定要有標籤。你唔好以為咧畫咗啲大試管一個咁嘅兜兜咧，我就知道係大試管，我係唔知嘅。就等同於你畫個細胞一樣，畫完個圓咕碌咁樣嘅嘢，跟住啦指出嚟係咩嚟㗎？你都會畫翻啲標籤。如果唔係，人哋都唔知你畫緊個細胞核啦、個細胞膜啊、個細胞質啊，諸如此類。好似梁 Sir 直接俾咗個圖畫你先，係因為我想聚焦去講翻究竟點樣去諗呢一條題目出嚟嘅。因為梁 Sir 想集中去講翻點樣透過科學探究嘅知識去建構一個裝置出嚟嘅。咁對於每一個儀器，你要有少少嘅認知先啦。咁點樣去畫一個裝置呢？個關鍵位就係個 DV， 我哋要觀察啊嘛，觀察酶嘅活性。而點樣去觀察酶嘅活性啊？就係觀察翻喺整個反應嘅頭5分鐘，佢產生咗幾多嘅氧氣。咁所以我哋就要肯定，我哋整個裝置要有2個功能要做得到啦。第一就係收集到啲氧氣。第二就能夠去量度得到究竟有幾多氧氣產生得到啦，所以我哋要選擇嘅呢就係一個適當嘅儀器，而今次呢個儀器就係儀液管，呢一支呢就係儀液管啦，佢有刻度嘅。咁而家啦，呢2個裝置呢都係將個儀液管就倒轉擺。嗱，留意下啦，喺中一呢大家學咗噶啦，一支真正嘅儀液管梗係有刻度啦，係咪？但由於啦，當我哋畫圖嘅時候呢，係一個縱切面。咁所以咧，你係見唔到啲刻度嘅，你只見到一支光禿禿嘅管嘅啫。咁而家啦，我哋先睇呢個裝置點樣符合呢個條件啦。咁首先啦，先睇下呢個裝置啦。喺個大試管入面同埋成個儀液管咧，都已經擺晒個 reacting mixture， 即係撈埋咗雙氧水同埋過氧化氫酶噶啦。咁你就成支管啊，只要你有呢個 reaction 嘅 mixture 咧，佢都係會進行呢個反應，都會有氧氣嘅產生。大試管所產生嘅氧氣咧，就經過呢支管噗嚕噗嚕噗嚕咁樣上咗去啦。而喺儀液管咧，其實入面都有雙氧水同埋過氧化氫酶噶嘛，呢度都會有氧氣產生嘅，所以漸漸就會有一個高嘅氣壓出現，就會將啲水撳咗落去，你就會見到個 reacting mixture 嗰個 level 啊就會向下啦。咁其實即係見到啦，個水位咧就會向下喐咗特定嘅位置，咁你就見得到啦，究竟有幾多氧氣咧係產生咗啦。去到右手邊同樣原理啦，當呢個 mixture 氧水同埋過氧化氫酶咧，佢又係會產生氧氣。當啲氧氣積聚咗響樹咧，佢有一個高嘅壓力，就會將呢一個墨水咧就推咗上去啦。你又可以睇下呢個墨水咧喐咗幾多嘅刻度啦，從而咧就判斷翻有幾多氧氣咧係產生咗出嚟咯。好，咁提多一樣嘢啦，呢2個裝置咧都有一個唔同嘅，就係左手邊呢個裝置咧有個夾夾嘅。咁其實呢個夾夾咧就係唔俾啲氧氣離開啊，啲氧氣出咗嚟噶，但係當呢度積聚咗嘅時候咧就係一個高壓咁啊撳咗落去咯。但係右手邊唔同喎， 我哋唔需要個夾嘅，係因為我哋想喺個墨水嘅左右兩面咧，都形成一個唔同嘅氣壓。大試管入面嘅混合物，佢哋製造咗氧氣之後咧，呢度就有氧氣嘅積聚，氣壓就會增加。氣壓增加不特止，仲要高過喺墨水嘅另一面，即係大氣壓力。呢個粒墨水咧就會向上喐啦。今日仲有第三個裝置嘅，同樣原理嘅啫。喺呢個大試管入面嘅混合物，雙氧水就俾過氧化氫酶拆解變成氧氣，氧氣產生，呢度有高嘅壓力，啲氧氣有路走啦，經過膠管走，去到啲水嗰處咧，我哋會見到嗰啲氣泡嘅，噗嚕噗嚕噗嚕，氧氣咧就會俾個量筒去收集咗。而當呢度聚集咗氧氣之後咧，氣壓就會增加，就會將個水位向下撳。咁量筒都係有刻度噶嘛，咁你就會見到啦，究竟有幾多水俾人逼咗落去，即係啦，呢度有同樣體積嘅氧氣咧出現咗啦。好，又嚟到一點出發啦。今次嘅題目咧就由過氧化氫酶咧去開始嘅，講嘅咧係三樣嘢：新陳代謝，跟住啦影響酶嘅活性嘅因素，同埋科學探究。先講新陳代謝啦，兩款類型啦，一定要識啦。分解代謝、合成代謝，不同例子大家要知，因為無論係 MC 咧定係短題目都可以問你啦。蛋白質嘅合成啦、脫氨作用啦、複製 DNA 啦，邊一啲係分解代謝，邊一啲係合成代謝，你分唔分得清呢？呢個啦就係影響酶嘅活性嘅因素。喺我哋書本我哋學過溫度啦，同埋酸鹼值啦，咁當中都有啲 keywords 嘅，就係動能啦，就係當初溫度好低嘅時候啦，酶嘅動能啦，同埋受質嘅動能啦，都係好低，即係佢哋喐得好慢，咁就少啲機會去分到呢個 M-side substrate complex 啦。但係啦，當溫度慢慢增加，佢哋嘅動能開始增加啦，咁咪大啲機會 form 到 M-side substrate complex 啦。而當個酶佢喺一個高於佢嘅最佳溫度嘅地方去工作嘅時候咧，其實佢漸漸咧就會變性。亦呢個情況咧，同極端嘅酸鹼值都一樣嘅。當個酶係一個唔處於佢最佳 pH 值嘅話咧，個酸鹼值嘅環境做嘢咧，其實佢個活性位置都會變形，亦即係變性，就唔能夠同個受質 form 到 M-side substrate complex 啦。跟住啦，去到科學探究，變量唔使審噶啦，條條都會問你噶啦。今次 IVDV 講晒俾你聽 ，CV 我講咗一個俾你聽啫，就係雙氧水嘅體積。咁仲有冇其他嘅控制變項呢？你噏唔噏到俾我聽呢？喺留言嗰度打翻俾我啦，睇下你識唔識咯。第二啦，就係畫翻個 set up 啦。今次點樣畫 set up 呢？記緊嗰個邏輯咯，就係透過個 DV 去俾你一個提示啦。因為 DV 就係你觀察嘅嘢。今次我要觀察產生幾多氧氣，自不然就要揾翻一啲適合嘅裝置去用啦。215 question 7 is about the experiment of M-side. First of all, we need to talk about two chemicals. Hydrogen peroxide is the byproduct of some metabolic reaction inside our body. If we were allowed to accumulate, it would kill our cells because the hydrogen peroxide is toxic. It will kill our cell. Eventually, it will kill us. Fortunately, we have an enzyme called catalase, which speed up the breakdown of the hydrogen peroxide into water and oxygen. So you can take a reference of this equation. And in textbook, we have already learned about this enzyme. This enzyme, we can find it in the plant and animal tissues. For example, apple tissues, potato tissues, liver tissues, and also the meat. And then what about the scientific investigation? We are going to investigate the effect of the temperature on the activity of the catalase. So you can see that the steps 1, 2, 3, 4, 5, that's the basic draft procedures of the investigation. We shall talk about the procedures in details in part B. And for part A, we need to take a look at the first procedure. Extract the catalase from an animal organ. So in part A, we need to suggest an animal organ in which catalase is present in great abundance. That means a lot of the catalase. And from which of the enzyme can be obtained. Because this organ contains a lot of catalase, so that's why it's easier for us to extract it from this organ. And we need to explain why this organ has such much catalase. So from the textbook, we have already learned that the liver tissues, it contains a lot of catalase. It checks the concept that we need to recall the role of the liver. So liver is a multifunctional organ. In the digestion topic, we learned that it can produce bile. In the homeostasis topic, we learned that it can regulate the blood glucose level and store the glycogen. And for this question, we talk about the detoxification. It can deal with the mild toxic substance, for example, alcohol and drugs. We also have the scientific investigation question about the alcohol and the beetroot. So the alcohol, it can dissolve, it can destroy the cell membrane. Therefore, it can also harm our cells. And for the drugs, it can kill the bacteria, kill the virus, so it may be also harmful to our cells. So remember that not every toxic substance can immediately kill us. There will be a lethal dose. So that's why that's not the matter of yes or no. The toxic substance, it must be harmful to us. But the idea, it should be more or less. How much or how less is the toxic substance can really kill us. If you only intake a small amount of that particular substance and then it can kill you, this substance, it will be extremely toxic. So that's why we need to recall that the animal organ is the liver. Because liver itself, there are so many metabolic activity take place. And hydrogen peroxide is the byproduct of the metabolic reaction. So for the liver itself, it must contain a lot of catalase to deal with such mild toxic substance. Or we can say that the liver is the organ for detoxification. So for the part B, we need to deal with these 5 steps. So the student has missed out an important step in his draft procedure. What is this? And explain the importance of this step. So let's take a look at these 5 steps. First of all, we extract the catalase from the animal organ and then we mix the hydrogen peroxide solution with the catalase extract and then replace the mixture at 0 degrees Celsius and measure and record the volume of oxygen gas released in the first 5 minutes and repeat step 2 to 4 with the water bath set at 20 degrees Celsius, 40 and 60 degrees Celsius. So it seems like that all the 5 steps, they are very normal. We mix the enzyme and the substrate together and put them in the particular temperature. Obviously, we can investigate the effects of the temperature on the activity of the enzyme. However, this step, there is a loophole. But before we talk about the loophole, we need to do some revision about the scientific investigation. In every scientific investigation, Mr. Leung will talk about the IV, DV, CV for you. First of all, for IV, what we investigate, we are investigating the temperature of the reaction on the activity of the catalase. So that's why IV is the temperature of the reaction. And for the DV, it is the activity of the catalase. But how can we observe it? We measure the volume of the oxygen gas released in the first 5 minutes. For the control variable, we need to keep it constant in different setups. So there are 4 setups and different temperature. I give you 1 CV first, volume of the hydrogen peroxide solution. What about others? Can you name some for me? We need to think about that. We are investigating the effect of the temperature on the activity of the enzyme. Therefore, the critical skills, the critical steps is that we need to make sure the enzymes and the substrate, they are at the same temperatures before mixing them. So just like in this situation, let's say the hydrogen peroxide solution is already at the 0 degrees Celsius. But what about the catalase extract? It is not at the 0 degrees Celsius. It's at room temperature, 25 degrees Celsius. And then we mix them together. So with the final mixture of the catalase extract and the hydrogen peroxide solution, mix it together and immediately it will drop to 0 degrees Celsius. It must not be. Because the catalase extract is 25 degrees Celsius and mix it with the 0 degrees Celsius solution, it will be higher than 0 degrees Celsius. And you may say that, no, Mr. Leung, if we mix them and then we put in the 0 degrees Celsius for several minutes, finally it will reach the 0 degrees Celsius. Yes, I agree. However, before it really reaches the 0 degrees Celsius, the reaction has already begun. And that means your experiment is not accurate. So that's why the critical steps is that we need to place the test tube into the beaker of the water at different temperatures for around 10 minutes before we mix them. So that's why we make sure that both of the catalase and the hydrogen peroxide solution, they are at 0 degrees Celsius, 20, 40, 60 degrees Celsius, and then we mix them together. and it is the importance. Ensure the mixture has already reached the set temperature at the beginning of the reaction to make the whole experiment is more reliable. And for part C, and you are provided with the following apparatus and material, and you need to draw the setup. This question is a bit different from the experimental questions before. In other experimental question, it may ask you that the setup is already like this. And then what is the importance of this setup? Why do we need to set up like this? Or a particular steps, why this step is so important? But now it asks you to draw the setup. And I attach the setup to you already. Because I would like to focus on the critical skills. How can we think about the setup step by step? And there are two critical skills we are going to talk about. First of all is the scientific investigation, and the second one is the scientific drawing. So for this part, I would like to remind you that just like the biological drawing, after you draw the setup, after you draw the cells, you need to do the labeling to tell me what is that structure or what is that apparatus of the material. So for the scientific investigation, what is it about? How can it lead us to draw the setup correctly? First of all, we need to recall the DV, dependent variable, which is the activity of the catalase. It is what do we observe. We measure the volume of the oxygen release. That's why we need to make sure that your setup can fulfill the two criteria. First of all, it can collect the gas. Secondly, it can measure the volume of the oxygen gas release. Therefore, we need to choose the suitable apparatus, which is the pipette. So you can see that there are scales on the pipette. And then we put it in the setup, and then let's see how does it work. So for these two setup, we put the pipette in the upside down manner. One thing I would like to mention is that for Form 1, you have already learned that how can you draw the setup. Remember that it is the vertical section. So that's why you cannot draw the scale on the diagram. So what about these two setup? What is the mechanism behind the setup? So let's see the left hand side first. Reacting mixture contains the hydrogen peroxide and the catalase. And then once the reaction starts, oxygen gas will be released. So the gas released and then they will rise to the top. And the reaction mixture, they also present here. The oxygen gas are also produced here. And there is a clip here to prevent the air from escaping. And it will push the reaction mixture level downwards. And then we can take a look at the scale. How much of the reaction mixture are pushed downwards. And then for the right hand side setup, the reacting mixture, it also produces oxygen. And the oxygen will accumulate here. There will be a higher pressure here. And you can see that there is no clip on this side because it allows this part connected to the atmosphere. Therefore, on both sides of the ink, there may be pressure difference. The boiling tube, there will be a higher air pressure due to the oxygen production. So that's why the air pressure will be higher than that of the atmosphere. So the ink will be pushed upwards. And then we can see the scale again to measure the volume of oxygen gas produced. And for the third setup, the same concept. Oxygen released to the boiling tube and the oxygen will accumulate and there will be higher air pressure. So the oxygen gas will travel along the rubber tubing to the measuring cylinder and you will see some air bubble here and the measuring cylinder will collect the gas. And in this part, the gas will accumulate and there will be a higher pressure and it will push the water level downwards. And then you can see the scale of the measuring cylinder to let's see how much oxygen is produced. So let's talk about the curriculum mapping. This question starts from the catalase and there are three concepts we are checking. Metabolism, the factors affecting the enzyme activity, and the scientific investigation. So for the metabolism, catabolism, anabolism, can you recall any example? Protein synthesis, deamination, DNA replication, can you recall the concept? And for the factors affecting the enzyme activity, temperature, pH values, we learned it from the textbook. We need to recall about the kinetic energy when we are talking about the temperature or when the temperature is so low, the M side, the substrate kinetic energy is also low. So that's why there will be lower chance for them to combine together to form the M side substrate complex. And what about the M side is working at the environment which the temperature is higher than the M side's optimum temperature? So the M side will denature. That means the shape of the active site will be distorted. And also for the pH value, same idea. If the M site is working in the extreme pH value, which is away from the optimum pH value, so the M site will also experience denaturation. And for the scientific investigation, what about the variables? IVDV I have already given you, and for the control variable, I just gave you one of them. So can you recall any other control variable? Leave your answer in the comment section. And what about the setup? It may ask you the importance of the setup, and it can also ask you how can you establish the setup. And the key idea is that make good use of the dependent variable. What do you observe? It will tell you how do you observe.